Okay, this is part one of a two-part lecture on science misconduct titled How Not to Do Science. So as you're all training to be scientists, what you will find is that there is a lot of irreproducible science out there. A lot of science that is published in peer-reviewed journals and is just plain wrong. And what we're going to cover in these two sessions, these two videos, is why there is so much rubbish science out there and so many studies that can't be reproduced. So the scale of the problem was identified in 2012 by a company called Amgen, who tried to reproduce many of the key findings in 53 key studies. And if you go to the PowerPoint version of the slides and click on this link, you can read up all about this problem. But what they found is when they replicated these 53 key studies, they found that only 6 of them were still standing. The rest of them were effectively disproved by the company's own research, saying that those research publications were false. And we're going to look at the sorts of things that can lead to a false result being published in the scientific record. One problem with this study is Amgen never actually released which of the studies were tested and which ones were successfully confirmed and which ones were falsified. This has been a very contentious issue. Lots of people would like to know which studies were disproved. So throughout these two sessions I'm going to cover the sorts of things that causes things to go wrong in science, and this can be scientists unintentionally presenting non-representative data, and this can be down to poor logic. It could be scientists attempting to effectively prove their hypothesis rather than test it. It could be genuine errors, sometimes unexpected controls being needed, and honest mistakes. And I'm going to go through all examples of all of these things. Sometimes it can also be down to poor tools for research. So in biosciences we all use cell lines and animal models, and these do not necessarily represent human disease. So if you find some preclinical work in these things, it might not translate through to the human situation. One of the key things that we come back to time and time again is that some antibodies do not work, and I'll show you an example of my own work with an antibody that didn't do what it was supposed to do. And then in the second session we'll cover deliberate science fraud, so fake data, fabricated data, image manipulation. So these things here are scientists not really doing as they should, not thinking as clearly as they should, or using tools which are not as good as they think they are. Whereas here we're dealing with deliberate fraud. So there are many different types of research misconduct. Fabrication is the most obvious thing. You just make up some data that never actually was acquired. Falsification of data, similar to above, but altering data to slightly represent something else, maybe cutting corners, omitting vital controls, generally being a lazy scientist, which happens quite a bit. Plagiarism is a real problem, where people effectively copy and paste out of one paper and create a new paper. And this is particularly problematic when people steal people's ideas. And the whole science peer review process involves scientists reading publications and grant applications and then commenting on whether they are worthy of publication and funding. And this process can break down where the peer reviewers who are supposed to be reviewing that work steal the work and publish it themselves and pretend that they had done it. And this happens. There's a concept that is particularly relevant to the biosciences called p-hacking, and this is where you get a set of data and you want to get to the magical p of less than 0.05 because that's statistically significant if you're a lazy scientist. And you can tweak the data until you get to 0.05, and therefore you can use those words, this data is significant. So I'll take you through p-hacking, but what we find is there's a disproportionate number of studies out there with a p-value of a p of 0.049, and that is a strong suggestion that that data has been tweaked because very few people publish data with a p of 0.051. We call this p-hacking. Another thing that can come up in research misconduct issues is ethics. Maybe you're performing procedures using tissue samples that you don't have ethical approval to have obtained and work on, or you're using data without permission of the person who generated that data. Every now and again we get MSc research project students who publish the data from their final research project without informing their supervisor. That is research misconduct. There are conflicts of interest, so some people may work for a company that has a financial benefit of that result being published and being correct. So a conflict of interest like that should always be declared so that we can take that into account when reading the publication. Sometimes people are funded directly by a company, and this has happened on many occasions. A company will fund a person to do a piece of work. They will get data that the company likes and therefore subsequently get more and more funding. So this would be a classic conflict of interest. And then this is probably the commonest one that a lot of scientists fall into this trap, is manipulating incidental findings and pretending that whatever you found was the thing that you were looking for all along. So you set out to find out whether A results in B. You actually find that A causes C instead, and then you report that you hypothesize that A causes C, and here is the data that supports your hypothesis. And although the data might be true that A causes C to happen, it's not the correct way of reporting that. You should always report what you set out to find, and that if you found an incidental finding, it should be reported as an incidental finding. So most of the unconscious and unintentional research misconduct generally comes from poor oversight by junior researchers, by the research leads. So basically letting junior scientists go away, do things, and then all the results that they bring back to them at the lab meetings are taken on face value and accepting that data without question. Quite often we find poor record keeping when there's research misconduct, hence we really push you to keep tidy lab books. Lab books should always be dated for all of the experiments that are done and then double signed by a supervisor on a regular basis. And what we find is that when research misconduct has occurred, quite often the lab books go missing, which is always a little bit suspicious, and that's usually a sign that the experiments had not been done properly in the first place. So this is sort of, more at the severe end of almost deliberate misconduct here. But we can also see poor reasoning in most scientists. No matter how good a scientist you are, there is often unconscious bias and what we call logical fallacies. And these are some of the more common ones: confirmation bias, appeal to authority, false cause, and the black swan hypothesis and falsifiability, which we will go through. So confirmation bias is a very common issue in science, and this normally occurs because the professor, the head of the research group, expects a certain result based on their own hypothesis. The junior researcher goes away, gets lots of results, and only one of those sets of results matches the professor's the professor's hypothesis, and so the junior researcher presents the one set of data that matches the professor's hypothesis in the lab meetings, and that's the data that gets published. And the 4 times that they did it and it didn't work get quietly ignored. Now, this can be a common occurrence on junior researchers, and especially with research supervisors who bully their junior researchers into getting the data that they want to see. So this is summarised by this little cartoon. I did 6 experiments, only one of them supports our hypothesis. Well, let's submit that experiment to the grant proposal. What about the other 5? Let's put it down to human error. And you hear this all the time, and it should always be challenged. Now, one important lesson for you if you end up in a research lab is always find out whether it's okay to disprove your professor's hypothesis. And my mentor, when I was a junior scientist, always told me to try to disprove whatever the current hypothesis was, and if you tried your best to disprove it, then if the hypothesis was still standing, well, you never know, the hypothesis might be correct. And that's a really good way to think about science. So related to the previous one, appeal to authority is a major fallacy in the lab, because the general assumption is that the professor is always correct, The professor is more senior, the professor knows what they're doing. So therefore, if you get data that disproves your professor's hypothesis, well, the professor might not be happy about that because they are in authority, they know what they're doing, you must have done something wrong. So the two, confirmation bias and appeal to authority, are often closely linked because the professor came up with the idea, the professor expects you to get the right results which supports their hypothesis. If you don't, well, it must be you that's done something wrong. Okay, this is also a very common issue within science, the false cause, and this is where poor reasoning makes you think that because A, the presence of A, which could be a result, correlates with result B, therefore A causes B. And what we'll study in part 2 is the work of Andrew Wakefield and the MMR vaccine and measles and autism, and we'll see classic examples of false cause going on there, where you think that the correlation of some events, one must cause the other, and we will find out that that was just one of the things that were wrong with Andrew Wakefield's work. And this cartoon summarises what I'm always saying to my students: repeat after me, correlation does not imply causation. Just because two things correlate, it doesn't mean that one of them causes the other thing to occur. And this is a classic example of poor logic and poor thinking, and it's not biosciences, but it really illustrates the issue, and it's under the heading of survivorship bias. And this is a representative image of a plane. It's from World War II, and it shows the military, the damage to these planes when they return from going out on missions. And somebody looked at these planes and thought, those bullet holes seem to be clustered in certain particular places. We should use this data of where the bullet holes are to find out where we should improve our shielding. So when you look at that picture, you see where the bullet holes are, you have to think, where would you put extra shielding to maybe improve the survival of these planes? So have a little think about that for a moment. Keep thinking about it for a few more moments. So in this study, a whole load of work was done, and there was someone called Abraham Wald who realised that if you're only looking at where the bullet holes are in the planes that return back to base, you have something called survivorship bias. So basically, some of the generals were saying, oh look, the ends of the wings seem to get shot at quite a lot, let's put more shielding there. Whereas what Wald realised was that because of survivorship bias, these are the bits of the plane that can be happily shot at, and it will still make it back to base, whereas if you get a bullet hitting there or there, it won't make it back to base. So this is what we call survivorship bias, and this type of bias occurs in lots of experimental systems much more widely than you think. So for you bioscientists who are listening to this, you may well be thinking about the genome. There is survivorship bias within the genome. So the genome accumulates mutations due to radiation and agents that damage DNA, and they don't occur equally throughout the genome. They occur very, very rarely in very important genes, Hox genes, histones for example. If you get a mutation in one of these, that's not compatible with life, so you very rarely see any changes to Hox or histone genes. Less critical genes can tolerate mutations and actually help drive evolution. So you think about things like hemoglobin, sickle cell trait and resistance to malaria, that type of thing. Changing one amino acid, although it confers some degree of disease and weakness, it will also confer some sort of benefit. Whereas non-coding regions, which don't really have much of a function, readily accumulate genetic damage at a much higher rate than a less critical gene or a critical gene. So this is an example of survivorship bias that's easily visible and understandable in a bioscience setting. Okay, it seems that we keep going completely off-piste from science, but bear with me. I'm now going to talk about another bit of bias, and I put this under the heading of the black swan hypothesis, and many scientists will talk about the black swan hypothesis. So for those who don't know, especially if you're new to living in the UK all swans are white. We have 3 different species of swans: hoopers, Bewicks and mute swans. These 2 visit the UK in the winter. These float around on the local parks all year round. They're all white. When they're adults, they're all white. So I want you to have a think, and pause the video now, have a think about how you would test the hypothesis that all swans are white. So pause the video, write down your thoughts. So I've done this with my research students, and I've got all of the right and all of the wrong answers. And scenario 1, I've had this response. Student goes out and searches for as many white swans as they can find. They come back and report to me, I've seen hundreds of white swans, therefore all swans are white, hypothesis proven. Is that correct? A different student comes back and says, well, what we've gone out and we've looked at everything, looked in all of the places where you'd expect to find swans, and we took a very accurate note of all of the swans that we saw, and indeed everything that we found were white, but we were specifically looking for things that were not white. So they come back and say, we found hundreds of white swans, we failed to find anything other than the white swan, and therefore the hypothesis is supported. Not proven, but supported. Now in the third scenario, the student goes out searching for all swans, looking for swans that are non-white, still taking note of all of the swans which are white and ones that are not, and is lucky enough to come across this thing, which is a black swan. So in this situation, the hypothesis is completely and utterly disproven. The initial hypothesis was all swans are white. Scenario 1, the student only looked for white swans and only saw white swans. Scenario number 2, the student looked for black swans, didn't find any, so the hypothesis was supported. In this case, scenario 3, we've found an example that completely and utterly disproves the hypothesis. And this is what we do in science. We spend our time looking for effectively what we call black swans, things that will disprove our hypothesis, not things that will help to prove our hypothesis and make our professors happy. So if you want to look at the work of any science philosopher, probably the best one to look up is Karl Popper, who really coined the falsifiability as a research philosophy, where hypotheses are proposed, like we've done here. We propose the hypothesis that all swans are white, and we have designed experiments to go out there and actively disprove our own hypothesis. And if we can disprove the hypothesis is disproved. If we can't disprove the hypothesis, then the hypothesis is supported. And that is the way we tend to do science. So the problem with science at the moment is it's littered with these white swan experiments where scientists come up with a really good idea and then spend their lives generating data that supports their hypothesis and ignore anything that passes by them that might disprove their hypothesis. Whenever something goes against that hypothesis, they find a reason why not to use it, and therefore you end up with false data entering the science record. The strategy should always be do all the experiments to disprove the hypothesis in as many different ways as possible, and if you can't disprove your hypothesis might be correct. And it only might be correct until someone comes along and properly disproves it with a better experiment. So what I'm going to do now is take you through some data that has been generated in my own lab, which comes under the heading of irreproducible science. And this really comes under the heading of poor models, poor materials or reagents that result in false observations. Now, for the biologists amongst you, you'll know that antibodies are widely used in bioscience to detect specific proteins, and we tend to use monoclonal antibodies that recognise a single epitope of a single protein to perform our analyses, and then we can use those antibodies for things like Western blotting, ELISAs, immunohistochemistry. Now, we did a whole program of work that relied upon a particular anti-OPG antibody. OPG is called a protein called osteoprotogerin. On the Blackboard version of the slides, there was a lot more data that we did in our lab that I'm not going to go through on this lecture. You can have a look at it yourself. But basically, to cut a long story short, we've been using an antibody to detect osteoprotogerin, and we were getting some results that were a little bit odd and didn't quite agree with the rest of the academic literature. So this experiment is a Western blot. This is where we separate proteins based on their size and then detect specific proteins with an antibody, in this case to OPG. And we had cell lines that produced lots of OPG. This is MG63 osteosarcoma. They produce loads of it. That's been well known for years. And this is our tumour cells that seem to produce the wrong size OPG. And we saw this, and we kept seeing this in lots of different cancer cell lines, and we were finding OPG where it shouldn't be. But our work was based around this OPG protein potentially stopping an anti-tumour agent called TRAIL that we were also working on at the time. And this anti-tumour agent TRAIL couldn't work if OPG was present, but we were finding this OPG protein in a different form based on our Western blots. So could this be a different version of OPG? Could it be a splice variant? Could it be something else going on? Could it be a degradation product? So what we set out to do was prove whether or not that antibody was detecting OPG. So the approach we took was a two-dimensional gel electrophoresis. So you separate the proteins on the basis of their PI, and then subsequently separate them on the basis of their mass. So initially you separate on the basis of charge, and then you're separating them on the basis of mass. So it's 2D gel electrophoresis, and then we used our antibody to find out where on this two-dimensional gel we could find our protein. We found our protein as two little spots, and this is from the cancer cells here. So there's our protein. All we need to do now is find out what that protein is. Ask the question, is it OPG or is it something else? And what we found was that the protein that this antibody was detecting was not OPG, it was carbonic anhydrase type 2, a well-known tumour marker. So we're buying in an antibody that says anti-OPG. It is detecting OPG, and we know it detects OPG because that's OPG, but that is not. So we've actually got an antibody that's detecting two different proteins at the same time, completely messing up our analysis. So to be absolutely sure that this was right, we did all sorts of experiments to find out what was going on. We used recombinant human carbonic anhydrase, and we compared that to the cancer cell lines. We used recombinant human osteoprotegerin and compared that to the cancer cell lines, and found that this antibody could detect both proteins even though the vial said it should only detect one of them. Unfortunately, us and lots of other people had used this antibody extensively for immunohistochemistry, which is cutting sections of tissues, staining with the antibody and trying to locate where the protein was. And what we found is exactly the same staining pattern for the anti-carbonic anhydrase antibody and the anti-OPG antibody. These two staining patterns are very, very similar. So this suggests that everybody who's used this antibody, their results are wrong. Now there is a subtle difference between these two, is that the anti-OPG antibody also picks up some staining in the extracellular matrix from the stromal cells, which is actually where we thought the OPG was coming from. That was not picked up by the CA2 antibody. So this is what we were actually looking for, but we're also finding all of this as well, that everybody else was reporting this as OPG when it was actually carbonic anhydrase type 2. So the question is, what do you do in this situation? We've generated data, we've put it in our own papers. Other people have generated entire studies absolutely dependent upon this antibody, and that data is out there on the public record, and it's wrong. What do you do? And in science, there are a few different things you can do. You can publish a retraction, so that is where you completely withdraw the papers that have been published and state the reasons why you've withdrawn that work. And if it's down to science misconduct, this is what tends to be done. If it's down to something that's, like this, where it's an antibody that we've gone to the effort of disproving it, then that seems quite harsh because of the severe negative effect on the researchers' careers. Retractions are seen as a slur on your CV. So what you can do in our case is we cited all of the papers that had used the antibody in our paper, including one of our own studies, and in that paper we clearly stated all of the studies which had used this antibody where the results were possibly not quite as reliable as we thought they should have been. And that, from our point of view, we thought that was probably a really good way of dealing with the issue. It wasn't our fault. We were the ones who actually identified that the antibody was wrong. We probably shouldn't be retracting our papers, but we should put a note to say that we have subsequently shown that the OPG staining might not be as correct as we think it is, but the same is true for everybody else's work. And then what we also very rapidly did straight after is publish a study showing that OPG modulation didn't necessarily do what we originally thought it was doing, and we did a whole bunch of very complicated studies to basically disprove part of our own hypothesis. So our own hypothesis said that OPG prevented trail. What we showed is that in certain experimental situations from tumour cells, that was indeed not true. So I think what we've done here is done our best to set the record straight when there's data out there that could well be misleading. Okay, so that's the end of part one. I'm going to do part two up shortly, but before you watch part two, I want you to have a look at some of these links here. There's a link here to Retraction Watch. This is an organisation that logs all papers that are retracted due to research misconduct or things going wrong. So have a click on there and have a look around the site. There's also another site called PubPeer, which is like a peer review of existing peer-reviewed journals. And here's a link to a paper that is on the OPG trail topic area. That's why I'm aware of it, because it's a paper that I had read and had doubts about. If you click on that link here, and you'll see some really nice example of research misconduct, and then you'll actually see what the journal's response was, which I think was pretty weak and, well, not very good basically. So have a look at those two links and see if you think that's a reasonable thing for the journal to do to deal with what's an obvious case of false data. And then the other interesting thing you might want to do is have a look at Retraction Watch on their Friday Forensics section. Click on this link here and you'll see some examples. Every week they put a new example up of research misconduct or poor practice. And they put up images and you say, okay, here's this image. We've just got it from peer review from a journal. We think there's something wrong with it. What do you think is wrong with it? And sometimes it's clear misconduct, sometimes it's honest mistakes, sometimes the conclusion is we need to ask the people who've written this paper to do an extra experiment to support their findings a little bit better. So have a look at those sites, and then we will move on to part two of the session, which I will upload next, which is all about deliberate research misconduct.